Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI-CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of a discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
Well, good afternoon. It's 3 o'clock, July 27th, 2022. I see Ulla, I see Brendan. That must mean I am in the PI-CC steering committee meeting. Fantastic news. We have a completely and totally packed agenda, including some great updates and a few very cool upcoming sessions. So I want to get started right away. So, the first quick update is that there is an Advancing Telehealth Beyond COVID-19 Act of 2022. Now that's a mouthful. I hope I got that out right. This was from the Rules Committee. They have previously extended some of the exceptions related to telehealth for 151 days, and currently a total assortment of strikeouts and revisions in this, I won't read it again, Act of 2022 will remove several of the geographic requirements. So this is a clear indication that telehealth will continue beyond the end of the public health emergencies, and it probably alleviates many of the national concerns for many healthcare systems that have implemented some of these regulatory exceptions, including some of the telehealth medicine practices. So this removal again is expected to pass the House and will result in a 2-year extension of the telehealth and geographic exceptions related to the public health emergency. Now, one thing that's of course very hard to read into, and I have not had any chance to discuss this with a legal or some else counsel who knows the act. It's of course a very complex set of things. However, it's not able to identify devices or diagnostics mentioned, but so that remains to be determined whether that extends into telehealth diagnostics. But it's a clear indication that the government as well as the House, when this particular act will be passed, have a clear vector in the direction that the telehealth benefits will continue beyond the end of the COVID-19 Act. Again, devices and diagnostics I wasn't able to find, but I know several of you have, with their various groups, some advocacy activities in that space, and this act that's going through is very likely helping. The next element that we have received a lot of questions about is Medufa and Valid. I know many of you have emailed back and forth, and there's basically two positions. One is, will Valid be part of Medufa or not? That decision hasn't been made, but there's a lot of very important and condensed information out there that I wanted to present in a neutral fashion. First of all, CAP, and I know several of the CAP members will be on this call, have provided a very comprehensive update of laboratory developed tests as well as the Valid Act. They have dissected it out, and CAP supports many of the provisions within the Valid Act, and you can stay informed by using the excellent resources from the various advocacy updates, and we provide this here. The information is, to my knowledge, available publicly on the CAP website, so please take that and read it through and educate yourself on these topics, but a definite pointer to CAP and their comprehensive work on getting the information together. To switch that a little bit is that one of the proponents of the Valid Act, Senator Burr, has provided a press release. If I'm informed correctly, Ulla will paste the link to his actual personal website. And several reports have surfaced that state that there's a so-called clean version of the user fee bill without any of the extra components, i.e. the Valid Act. This all remains to be determined. And to be absolutely neutral, I also want to present the counter proposal or the counter position, which is that there are two letters to Congress currently on Valid, one for, one against. We of course share both. This is not taking any position. This is just providing the field with the relevant information in a neutral fashion. So we don't have any updates of whether that will be in there or not. So that remains to be determined, but this is of course a quick sort of update that there may be regulatory changes coming. The next part was a little bit of surprise, and many of you on the call know we have a charter. The charter sets out the mission, vision, and sort of the ground rules for this engagement of this pathology innovation collaborative community. We pay utmost attention to, being compliant with antitrust policies, but we have received questions on how to join. On the website, there's a button, top right, you can join, it's free, and that's it. But for companies, etc., it's that charter that is often in discussion. So it's free, available, and the charter can be signed. So if it's not as if this is a closed club, apparently that was sort of misportrayed or misunderstood at some point. And if you want your symbol, your logo, your sign on the landing page, that is of course a possibility. And of course also non-profits can join. So this is not restricted to industry. That was apparently not clear. So we may need to specify and spell that out. There were a few questions, and one element was also from several people is a proposal: could we start a non-profit organization that could help and act as a facilitator? There may be some rules about that. There's a specific section within the collaborative community guide or guidance that the FDA released, what the roles and the functions of a facilitator are, but more on that to come. But I just wanted to reiterate that that is already accounted for in the charter. In the next couple of slides, there's an update, so-called FDA corner, because the FDA is of course very active, and we selected out of the many policies and guidances that are surfacing those that are most pertinent to this group. First, there's one document that we have received several questions about, which is the technical performance guidance. I won't read the whole title. I think to many of you this is known. This is June 2022 version, so it's definitely interesting. A lot of quantitative imaging guidance on this, so I think Brandon and the other colleagues from the FDA know this very well. If you haven't seen it, please take a look. The second, which is also just a few days old, this is in draft guidance stage. So this also means we can comment and provide our opinion about it. This is related to conducting remote regulatory assessments. So this is, there's two components here that are probably of interest. One is, of course, as a draft guidance, it illustrates certain novel concepts. And then the second part is that there are so-called regulatory oversight tools available. So I won't have time to get into this, but I believe Ulla will paste the link. Maybe check it out. There's a lot of elements on how the FDA will continue to serve the public by protecting it through so-called RRAs. And the tools for remote regulatory assessments are, of course, different than for in-person or inspections. So definitely an interesting component. It's actually very well aligned with the first part here that I mentioned about removing some geographic requirements. So clearly this is one of the manifestations of some of the regulatory exceptions during the public health emergency. The way that this is done, again, is through certain use of inspectional tools. And one of the resources that I hope Ulla is also pasting in the chat is a so-called catalog of regulatory science tools. Now, we spoke several times about MDDTs or other tools or other pathways that the FDA offers. Here we're talking about a catalog of regulatory science tools. So these would be those that are used for inspections or, again, remote regulatory assessments. So it is not far to think, or it's not complicated to think, that some of those tools that are used for assessment are probably very meaningful to read to understand what you would submit. So we provide this link, and of course the FDA provides this catalog to inform the public. So definitely an interesting and very good resource. The next resource provided by the FDA is an FDA drug safety podcast. Now, of course, we're here not interested too much in drug development, more in the biomarkers related to that. We get to that back in a second. However, as everyone knows, podcasts are a way to communicate relatively quickly and effectively, and it is extremely good. And I think it's a super great accomplishment that this podcast already entailed one of the potential drug withdrawals because of a risk of increased death with a recent approval. So this is very current, and there must be a production team behind it that's acting very quickly. So if you haven't seen it and you haven't decided what your next podcast is, of course, it's always great to listen to what the FDA has to say. Second to last part in the FDA corner is a certain update to the electronic medical device reporting system. And there are several coding resources provided. So if you have any unintended consequences and any other things to be reported, this is a new coding resource for it. And then somewhat related to coding, but in this case more as a standard. And I know that many of the people here are indirectly involved in breakthrough designation or companion diagnostics, but this is more for the NDC, that's the National Drug Code that you find on any regulated drug and or compound, that will change. And that's, of course, a huge difference and a huge issue when it comes to making sure that all of that works well. Now, I know we have a standards work group here. The reason why I wanted to bring this up is that, of course, there's a lot of documentation related to this National Drug Code labeling update. And when it comes to changing and switching standards, that's, of course, very important. For example, thinking about file format changes down the road for devices. So quick hint that that's currently happening with drugs. For the next two slides, it's my absolute pleasure. We worked on this for a bit, and I think this will be very, very valuable for many of you. So it's my absolute pleasure to announce that Dr. Brandon Gallis, you see him on the call, he will also provide a quick update in a few minutes, has agreed to do a total of 3 hours of online sessions, and the first one will happen on August 5th. That's next week at noon. I hope you all have time. This is noon Eastern time, on evaluating medical imaging devices and image-based algorithms with the clinician in the loop. This will be a tutorial on reader study designs and multi-rater, multi-case analysis. So if any of that is interesting, I think this will be a highly valuable session. And it will also be, of course, recorded, and Brandon has agreed that will be hosted on the Truthing group that he helps lead. Now, this is not only the one session, there's also a twofer. There's a 2-hour online session, again, on assessing agreement and reader reliability. So this will be a redux of a session from the 2023 joint statistical meeting. We hear about that something in a second, but Brandon will find, if I'm correctly informed, a partner. So this will potentially be an interactive session, so should also be fantastic. So if you can reach your mouse or your trackpad, give some thumbs up and some applause here for Brandon, and really a great topic, very important topic, and it's really cool. So thank you so much. All right. With that, I hand it over to you, and this is the Truthing and Validation Work Group update.
Yeah, I just wanted to, thanks for the invitation to present that work. It's something that we've, it's training that I've actually given to some groups here in the FDA, the reviewers in the radiology group, and it's about the science of these kind of studies that have the clinicians interpreting the images and how to aggregate that data and turn that into some endpoints that could be useful to understand the performance of a medical imaging device or a computer aid of sorts. And that second session that Joe mentioned, Siwen and Siwen is a colleague of mine here. We have a session at JSM coming up in 2 weeks, and we are going to try and recruit our fellow presenters, and there's like 5 talks all about reader variability and reader agreement. So I think some of my colleagues here have been asking to have a chance to hear those talks, and this would be a great environment to do all that. My updates, I don't think there's a lot of things to emphasize there, except for it'll be in the minutes. Posting a link to that first bullet. Just in case anybody hasn't heard, there is a transition plan for when the pandemic is officially over, for what to do with some of those enforcement policies that were different during the pandemic. And so if you're in that space, I think it's worthwhile to check out that guidance document. One of our collaborators has gone full-time from a fellowship to a full-time position at the FDA, and that means I have a fellowship opportunity, so that link will be there if you have some younger folks that are looking for an opportunity here. And then we are trying to find an FDA cleared scanner to help us scan our images for our research, as looking for a scanner as a service in some sense, and happy to hear. I've been having a hard time finding one out there in the community, and so that's why I'm asking here if there's one that you know of. I think that's mostly it. Is that it? I think that's most of what I sent you.
And welcoming a intern. We have the summer back for a second time. Yes. So we have Phoebe Chin on the call, and she is a rising high school senior and is assisting us with a lot of work this summer.
Right. I like to point out some of these rising stars in the community. She is young and looking at colleges soon, but she has been impactful on my research as much as a college student or a postdoc even. I'm impressed.
Wow. I see the FDA gets them young. That's the trick. I now I understand.
Train them up. Train him up.
All right, we move on. So we have some time in the end for questions, or we can go back. There's I'm sure you heard about it in the press. There was a big issue about baby formula supply shortage. We won't get into that, but as one potential source of that, one of the currently proposed changes, and that's an update, is the FSA Act, so the Food Safety Administration Act of 2022, that proposes to separate food oversight from the FDA. That would mean that it is the drug agency. So this new enforcement entity would be focused fully on food and would be a separate branch under HHS. Now, you may ask, why is this relevant? You can be for or against it, of course, as always, but the relevance for drug development is that some of this has intake, has good manufacturing practices, has a lot of assessments, and that relies a lot of intra-agency experience across food and drug assessment. And we leave it at that, but it's, of course, as you can tell, there's a lot of action in the field. The next update, which I'm sure you heard in various ways, is a controversy around certain data presented years ago about the Alzheimer disease and certain findings within that. Why we wanted to mention this is for two reasons. First, of course, it has some regulatory relevance related to disease mechanisms and some of the drug approval / drug development. But one thing that I thought was very, very interesting, and this is from a recent Science viewpoint, where they explain how a possible dispute about data copying, so meaning fabrication versus similarity or dissimilarity of molecular data, here in this case of bands. There was an interesting XY correlation plot done where pseudo-coloring of bands and the subsequent overlay, when done with very similar bands, you will get a good correlation, while if there is XY scattering slightly different, you end up with this, I don't know, butterfly wing looking or insect wing looking pattern of distribution of pixel intensity. Now that's a simple XY correlation. However, that was one element to assess, in a way, quality and or this question of this possible dispute. If you haven't seen this or if you haven't followed it, I think this part, of course, we couldn't take all the things here for this one screenshot, but it's basically an approach to assess similarity of data points. And of course, this is image-based, let's call it dispute clarification, and you can judge for yourself what that is. But I thought that this image-based approach is definitely interesting, because I know a lot of people are interested in this, plus some regulatory relevance. And then in this news and updates, we have helped the sepsis collaborative community, at least in the early stages, and you may have seen there was a series of great contributions. There was a couple series of at least 3 papers in Nature Medicine, etc., and then other algorithms published in part in JAMA, etc. So we come to this, but clearly the field of sepsis, a deadly and very hard to diagnose entity based on integration of data or a multivalent transcriptomic metric, is definitely on the rise. So one of the things that's of course mentioned in the papers is how could this be made available as quickly as possible, and we come back then of course how to make that available, safe and risk averse. So definitely an area to look out for if you're into data analytics and these complex components. There's a couple of resources we wanted to mention, and some are actually really groundbreaking, and I'm glad that you joined, and I hope you will have some additional insights and food for thought. The first one was released in 2019, and it is very interesting to see how this now overlaps with the guidance on software as a medical device. This in this case is the qualification and classification of software in regulation. So this is called a medical software device, or MSD, under European rules. But the interesting thing in this resource is that this guidance, in this case from the European Union, entails concrete decision trees that are constructed not so much from a risk and or functional perspective, but more from where and what regulations would apply. So in anticipation of the IVD regulation in Europe, this is a very important work, and if decisions are made, if there's AI involved, that's all included. So a definite interesting resource for various reasons. The second and somewhat related is a IMDRF, so that's the International Medical Device Regulators Forum, that releases very comprehensive and interesting documents. And one is related to online learning. Briefly, that doesn't mean it's on the web or anything online. That means that you deploy a system and it continues learning while you assess performance. And in this document, the machine learning enabled medical devices, they define key terms and definitions. So you see, for example, if you make changes to a machine learning medical device, they define cause, effect, trigger, domain, and effectuation. So these terms are very specific, but they're extremely helpful, especially when doing performance threshold or training data or batch sizing thresholds, exposure to new data, etc. So it's a fantastic concept that encompasses many of the learning strategies that are currently applied in the field of AI. So if you haven't seen it, please take a look. And if you work in this field, it's definitely helpful, for example, when writing a paper, so that you can defer to definitions that are coming from an international group rather than you making those up and leading to further confusion. The next element, which is also a toolkit, this is a toolkit for so-called inclusive deployment. I have to admit that this is from the DATACC, that's also a collaborative community. They have reached out. We tried to schedule a meeting to really help how digital health measurement products can improve. So clearly the field of telemedicine is expanding, and we wanted to find out whether someone in this group is an expert in this area to help maybe navigate this collaboration. So in case someone is interested and has seen or worked with this DATACC toolkit, it's definitely of interest for especially, probably point of care or even home testing. Very, very excited about the next tool, which is absolutely incredible. And I think this is a game changer in the field. So the tool is called Prodigy, and it's available online. And as you know, I pulled here an old article from Forbes that has illustrated that about 80%, so all this green here, I presume that that's intended to symbolize that most of the money goes into data cleansing and cleaning of data sets. So if you take that, the interesting part, refining algorithms, is actually just a tiny little part. And what this company and product designer, you see her here, have enabled to do is a completely different way to annotate. So simply put, you have a yes or no or eliminate button where certain phrases, of course they use dog, car, bike here, where you can illustrate whether the algorithm, while you try to get a ground truth data set together, is functioning correct. You see there's overlapping is accounted for. So you would click on dog, you outline the dog, you accept that. You would click on car, it moves automatically actually to car, you annotate car, you accept that, and then you move to bike, and then you have one image labeled. So this is tackling with AI. So as soon as you entered this, it starts using your existing data to pre-annotate the next image. So what's so radical about this is that they tackle with an algorithm 80% of the work that data scientists have to do. And this is effectively speaking a radical new way of doing data annotation. Now you can imagine what you can do with this in pathology to include or exclude certain things. So if you haven't heard about this, this is Prodigy. I think it has something to do with Spark on the back end. There's several people who are the developers, but it's extremely efficient by the way, also this is the image-based application. There's also a neurolinguistic, meaning a word or text and concept-based application. So Prodigy, please take a look. I'm sure if you do a lot of data annotation, this would be helping, and I think this is spelled like that on the website, but I'm not 100% sure. The next resource, which I think is also incredible, so the WHO has many, many projects. And one is that they want to establish certain care continuums. Now, this is obviously a big overview, but what this is, simply put, is to identify, in this case for breast cancer, and they're planning to do this for many cancers, different pillars that are absolutely essential to make sure that you have something like a care cycle of breast cancer patients. So in other words, if we zoom in here, the tissue sampling as one example is part of the diagnostic workup. But as the part of the diagnostic workup, you have to have a positive rule in for a breast abnormality. So in other words, they codify a little bit the various care pathways that patients walk through. Now this has, of course, diagnostic implications, but also treatment interval, which is pillar 3, but also cancer survivorship and outcome to actually help, of course, improve patient care, but also help inform sort of like evidence-based frameworks. And so it's definitely a great initiative. The Global Breast Cancer Initiative is the name, so GBCI. Definitely look out, since I know many people are looking for TILs and how to get TILs implemented, since that's potentially a predictive marker. In these pillars, those are pathology results that definitely inform in terms of the biomarker testing, for example, the treatment interval. And you can see biomarker testing is in the treatment pillar. So this is the cutting edge groups that implement this. And of course, coming from the WHO, it's international and it's usually collaborative and multi-stakeholder based. All right. We have one important project update. There's a lot of projects going on, but we didn't have time to go into every single project today because we still have a few more things to talk about. And the one thing that we did is, you may have seen it already, a session with many different people discussing the decision summary for the first FDA-approved AI algorithm, and the session was followed by a lot of questions. Some were posed in the chat throughout the session, and we didn't have time to answer those. So what we've done, and thanks again to the whole group and the all speakers and the representatives from the company, Mayo Clinic, CAP, DPA, and of course the FDA, and put the question and answers from the presenters in written format. And you can imagine with the FDA that has to go through the appropriate clearance processes. So all the questions, complete as they were in the chat, are provided in written format, so you can search on the project page. And I think that's a great resource, including, of course, all slides, the full talk, and the video. So as a resource, I think that is what we try to do and intend with this particular decision summary project. So if there are other questions or so, please feel free to reach out and or use the resource as appropriate. As many of you have probably heard, HER2 low. HER2 was one of the first predictive biomarkers ever. And HER2 low in breast cancer, there was a recent sort of breakthrough finding. It was a New England Journal paper. It was presented at ASCO, and that changed the testing paradigm in breast cancer a little bit. And similarly, there is a abemaciclib, I think that's how you pronounce it. Kia-67 is the biomarker, also in breast cancer. And these two projects have been proposed. One is probably more related to how to do this practically. The other one is probably more related to how to verify and quantify Kia-67 reliably within the cancer compartment of breast cancer patients. So both have direct treatment relevance, but require biomarkers that are reliable. So these will be two new project proposals. So in case you're interested in the HER2 low or the Kia-67 breast cancer project, please text Ulla in the chat or send us an email or send me an email. You know, we can include you, and those projects are upcoming. One project that made some good progress is the circulating tumor DNA project called Out of the Dark. We had one session that was, I think it was probably unintentional, but you can tell that the ctDNA field is booming, framed by two events from Friends of Cancer Research, who are also participating. I see Brittany here. Thanks for joining. There was one session by Friends called Expediting Drug Development by use of the ctDNA as an early endpoint. That is also the draft guidance that was discussed and analyzed in this session. And then there was another meeting, I think today at noon, about the patient advocacy for circulating tumor DNA as an early endpoint. So what I think we can say, if you take a look at the session, it was a very quiet session because the circulating tumor DNA draft guidance from the FDA refers, I forgot exactly, but to 11 other guidances. So this is trying to frame its way into the trial structure, the investigational device exemption framework, and it was great to have many people on the call and patient advocacy plus Friends of Cancer Research. So if you're working in this field, we hope that this is of interest. There were a few people that wanted to comment officially. I think the official commenting time is over, but I'm sure we will have a way that the information is captured and shared with the appropriate parties within the agency. I have to say a big thank you to everyone who has submitted papers. I'm learning a lot because this is extremely helpful to get papers delivered like that, and so I wanted to share a few of those, and there's a lot in digital path and in biomarker and some with regulatory science relevance, and I wanted to share those because it's quite impressive. So the first one is an interesting regulatory science paper that has done a review of the 50 drugs that were approved by the FDA's Center for Drug Evaluation and Research. So this is CDER, for a very interesting past observation, and that is which of those drug targets are supported by human genetic findings. And as you can tell by the title, 2/3 are. And what they relate and derive from this data is that apparently with a biomarker or with genetic founded evidence for a drug, there seems to be a better way, or the proportion of new approvals is higher, at least in a retrospective analysis. What that means is beautifully discussed. It is sort of course, if you see this is, I think it was published in Nature Biotech. There's a bio-business brief. I think you can get the connotation here that this retrospective analysis implies sort of an improved productivity, etc. So fantastic work. Thank you for sending. And if you haven't seen it, take a look. Plus, it's a good overview to see all the work that's been done. So it's probably fair to assume that this is on average one drug approval per week, which is quite impressive. So the next paper I also think is a phenomenal thing, and I see Joe Siren-Trappin is on the call. We have, thanks Joe, by the way, for producing sort of a disseminated token idea. We spoke about blockchain, and in this paper, the group established a blockchain-enabled indigenous data sovereignty framework where, as you can read here, the indigenous community members themselves can decide whether they limit internal or external collaborations by being able to give or withdraw permission to the data. So in this blockchain-enabled project, it empowers the indigenous data so that they can be used for what the community and the individuals themselves consider meaningful. Fantastic application of this technology in a genetic or genomic and rather sensitive use case. The next paper, you see just by the amount of authors what a incredible work this must have been. And what this is a decathlon. So this is not a grand challenge anymore. Consider it the Grand Olympics. So this is what can you learn when comparing across, for example, several medical segmentation grand challenges. The statistics is different. There are different tasks in the different, components. They also define mystery tasks, meaning that some of these were, sort of like just in the same image plane, but some algorithms detected something in there. Nature Communications, fantastic paper by Michaela Antonelli. So large group. I think all data sets are publicly available. So please take a look. And I don't know, this may be the beginning of decathlons in AI grand challenges. One paper directly relevant to whole slide imaging. I think a similar paper was published by Matthew Hanna's group from MSK. Here we're talking about the rapid deployment of whole slide imaging for primary diagnosis and search path at Stanford Medicine. Now please take a look here. The number of total slides produced in this department is on the order of 150 to 220,000 slides. And you see here that there's a rapid adoption. They provide project maps, adoption rates, and a lot of details about how they accomplish this. I think it's published in the archives, and I think it's freely available. So please take a look. And definitely, if you see the percent of slides scanned, that's a very great accomplishment. I think Ohio State has a similar adoption rate, but there are few institutions that can accomplish such a quick pivoting. The next paper by Kelly et al. Is something that we should all care about. This is spearheaded by the Evaluations Measurement and Assessment Department from ASCP. So this comes from a few scientists, Institute of Science, Technology and Public Policy at ASCP, so two ASCP-based collaborations, and they looked at data from residents and fellows about all kinds of components, as you see here, work-life balance. And so as it's a very complicated but well-designed study, and to give you an example of what they found, is if you look at hobbies, so let's pick a random hobby. Let's say your hobby is reading guidance documents from the FDA. What they distinguished is if you have a poor work-life balance that they measured otherwise, or you have a good life balance. So in a poor life balance, most of the residents and fellows would only be able to read, let's say 2 guidances a month. But if you have a good work-life balance, you can probably read about, 2 guidances a week. Now, this is, of course, a distribution. So this is just one tiny blip into that data. But I thought it's very interesting to see not just that poor or good work-life balance is good or bad, but they quantified it across a whole array of measurements. Job stress, the pressure that they feel to sign out cases, the pressure about academic excellence, etc. Other obligations, and here hobbies, personal interests. So it's a very interesting way to quantify the difference between poor or fair and good or excellent work-life balance. So great job. And it's a good resource to have, especially when people are down that you manage. Then you can show them, if you focus a little bit here, also for leadership, right? Pushing harder may not necessarily get the wanted or intended effect. I hope I'm not messing up the name, because this is Biagio Ricuciti. I don't know. I need help here. I'm not good with Italian, but this group published in JAMA Oncology in an original investigation, a phenomenal study that linked tumor mutational burden in PD-L1 blocking with a multiplex immunofluorescence assay to describe something that people have always expected, in this case a non-small cell lung cancer, that is, in tumor mutational burden high tumors, that there is truly a higher rate of PD-L1 expressing inflammatory cells, presumably due to the higher antigen load, and that is not the case in TMB low. But since this is using trial data and on treatment data, this shows that low TMB versus high TMB as an objective response rate is fundamentally different and very likely linked to the actual biomarker. And they did a phenomenal analysis with intratumoral, peritumoral, and at the invasion front, subspatial analyses. All data available, all quantified, and it's in JAMA Oncology. So fantastic work by Biagio and group. Of course, the author list is much longer, and as you can tell, the images that they provide are quite fascinating. There's a Nature publishing group, Digital Medicine, so that's at least as high as Nature Medicine, paper by Hutchinson Grimm, where they demonstrate how to integrate digital pathology and mathematical modeling for spatial biomarker dynamics. Now, what you see here that looks a little bit like Go is neighborhood mapping, where from cell detection, this looks like a geographic map, so still the tissue is spatially not resolved, into a neighborhood, or let's say closest approximate nearest neighbor technology, to have then a tumor immunity model where you can apply certain simulated biopsies was one of their use cases, to say, what is the chance of finding certain things and finding certain relationship. And this is published, as you can see here, in partnership with the Seoul National University. Fantastic work and a lot more statistics that I can't get into, but if you want to take a look, I think this approach I haven't seen before, and it's really interesting. And the math that follows is drawing from things that are known and established. The paper by Shinohara et al. Is a paper that proposes that you can accomplish substantial improvement of histopathological diagnosis by whole slide image-based remote consultation. Now, the study design is very interesting. You see that there were multiple rounds to identify. Ultimately, a consensus diagnosis by 6 senior pathologists was needed, and they're trying to accomplish sort of the impossible, which I think, Brandon, your MCMR approach is not exactly followed to the dot, but the data that they have would certainly be amenable. So we reached out and tried to schedule something probably after your meeting to see whether it would be possible to apply some of your methodologies to the data set. But they did a very good job in tracking everything, and especially focusing on the point, could you make the argument that whole slide imaging that enables remote diagnostic or remote consultation can actually improve? Their conclusion is it does, but requires probably confirmation from others as well. Now, speaking about resources, this is a paper, Marilyn Bowie, who we know from CAP, DPA, etc., and Louis Hassal, first author, and a long list of authors. If you look over that, many of those I've seen here as well, have put together the pathology education powered by virtual and digital transformation. This includes gross images, databases, slide scans. You see, this is sort of a collection of atlases and didactic resources, including some of the locations, and most of it available on the web. Those can be used for various purposes. So it's a great summary. I think this was also in the archives. Fantastic. Maybe you can share this with your trainees. I think this may be something. Maybe we can reach out through the trainees group of whether one of those authors would want to present sort of this meta atlas, and of course, serving as a ground truth, some of these elements here have been highly annotated. There's something additionally and very exceptional. I think the senior vice president of the Allen Institute for Brain Sciences, Honghui Zheng, has accomplished a single author cell paper entitled "What is a cell type and how to define it?" I mean, you have to have some stamina to accomplish that. And what she has done is a multimodal disintegration of all brain cell types of the mouse to reconstruct what that looks in a continuous spatial IT infrastructure across all the cell types. It's a great review on whether you want to be a splitter for entities or a lumper, meaning putting them apart or putting them together. And she discusses that, and it's a phenomenal piece of work. This is just one of the many figures. And, I have to admit, like, I haven't seen anything like that in a long time from a single author, of course, well accomplished, and congrats. But if you're in the field of spatial phenotyping, this paper is probably defining a new thing, especially when you want to define new cell types or define subsets of existing cells. There's several rules in there that come in very handy. A paper from a German group, Olaf Neumann, Albrecht Stenzinger is one of the second last author, I believe. I'm just mentioning this one here because a lot of interest is in FGFR2 inhibition in cholangiocarcinoma, and fusion detection is sort of tricky. And what's shown here is a fantastic overview of all the different assays that can be used to identify gene rearrangements, break-apart fish, imbalancing assays, sequencing assays, and also what each assay can detect and what it cannot detect. So this single slide here summarizes probably a whole day of our curriculum when we teach our molecular fellows. So if you have something to do with molecular diagnostics, this is a great single figure resource. I think it's a figure 2 in that paper. So amazing work. We also have received several additional resources. These are just simple pointers to it. I think this speaks to the use of the website, the PyCC website. This is from an author from the WHO, Mahmoud. He put together a practical guide for health researchers, and it is literally no overstatement. It goes from how do you pick your research field all the way to how to do statistical projections, planning, publication, how to effectively write, etc. And this practical guide for health researchers entails many of the regulatory paradigms. The book is a little bit older, but what's in it, it's absolutely not. It's written, of course, and published from the WHO, so you can probably see what that entails. The second, from the American Association for Clinical Chemistry, they released a point-of-care testing guide, how-to guide for the non-laboratorian on how to implement and how to perform point-of-care testing. There were a couple of other questions about whether we would tackle this. I have to admit I'm not a big point-of-care testing expert. Maybe some of the other people on the call are. Please reach out and let us know, because there may be some synergies with AAC and this particular group. Flagship Biosciences has produced a compendium about tissue-based pathology diagnostics for regulated applications, especially with hint to companion diagnostics. We were asked to share this. Of course, we share. And then probably related to the standards group, this is a BSI standards publication. And now we're talking really meta level. This is a standard for standards, the principles for standardization or of standardization. So the best equivalent to explain what this document is if there would be a guidance document on how to write a guidance document, but or a paper on how to write a paper. So we're somewhat removed, but again, this is was shared and should serve those people who work on standards fairly well. Right. I have to stop talking because I wanted to leave time for questions. The next month's steering committee is again 3 to 4 p.m., August 31st. And keep in mind before that, so next week Friday is Brandon's talk. We mentioned this before. So next Friday at noon. And with that, we can discuss any of the topics that we had here, or new topics, or old topics, or we can just chat. But thank you very much for your attention. And sorry it's going very fast, but there was a lot of interesting stuff to cover.
I'll ask a question, Joe. Are we gonna get any face-to-face meeting coming up? Because I have approval to travel to one, and if I don't get on planning that, they'll take that approval away.
Mm-hmm. So one thing that we always discussed was whether we should have a Boston Tea Party and symbolically throw a fake microscope in the ocean and say the time for digital pathology is here. Now, that was initially just like a fun idea, but it seems, with CPT codes emerging, approvals for, scanners, algorithms happening, that could be, a possibility to organize that. I don't know what people think about it.
I think it will controversially be mentioned in many conferences to come.
You mean that there are too many conferences, or?
No, I think that it would, if there was some picture or something, I think it would be controversially presented in many conferences to come.
That there was a Boston Tea Party?
Bad joke, never mind.
Now I got it, sorry. I don't know, what do people think? We could do it this year, October, or make it later. I mean, usually we have an annual meeting. So far we didn't have that because we have basically continuous meetings and projects, etc. But
maybe we could do a quick survey on that and collect people's ideas about yes or no, and then what month or even week would be good for something like that, or attaching it to another conference just to get more real.
I mean, USCAP
I guess
comes
I'm
to
not
mind.
pushing for it, but definitely if there's gonna be one, planning is critical on my side especially.
I mean, we have CAP conference, right? I don't know what other conferences we have. Vision, which I think is in Vegas. I think CAP is in New Orleans. Were you thinking about that, or?
We've been attached to pathology visions before.
Yep.
I think having a face-to-face session at Path Visions is a good idea for those who can attend. Pre-session or post-session, figuring out a day that works for everyone.
Sure, let's do a survey. Why not? I think that would be great. Brenton, were you and your team, Kate, Weiji, were you going to Visions? Were you planning to go there, or?
I think I have that on my calendar also. So I think those, I was hoping to combine those two would be nice. Save my division some money.
Brandon will be there. I will be presenting. Awesome.
Okay. Any other topics? So keep in mind for those who are representing companies, if you want to join, get your logo up there, that's an option. Participation in any project is of course always encouraged. If you have other project proposals, feel free to send those to Ulla or anyone in the other work groups or projects to learn a bit more. All right, then I think we can end on time.
Excellent. Thanks, Joe.
Thanks a lot.
Thank you, Joe.
Thank you.
See you soon. Bye-bye.